Hello and welcome to our video lecture on section 18.4, where we're going to continue our discussion of the adaptive specific immune system. And in this section, we'll talk about B lymphocytes and humoral immunity. Now, I've referenced this in previous sections, but this type of immunity is going to be mediated by our antibodies that are secreted by our B lymphocytes or B cells. So let's go through our learning objectives. We want to be able to describe the production and maturation of B cells, compare the structure of B cell receptors and T cell receptors, compare T-dependent and T-independent activation of B cells, and compare the primary and secondary antibody responses. And just as we did with our T cells, we want to talk about B cell production and maturation. So we start off the same way as T cells. We are going to have them form, or B cells that is, from our multipotent hematopoietic stem cells within our bone marrow. And then we have our lymphoid stem cells turn into lymphoblasts and stay in the bone marrow in order for them to mature, and then they can travel over to the spleen. So we do this in a series of 3 steps. We have our positive selection take place, where we are looking for normal functioning B cell receptors. And of course, if they do not function properly, we want to eliminate them by apoptosis. We can also edit or modify these receptors or have them go through the process of induction of anergy in the B cell. Second thing is we are going to do negative selection, where we are going to eliminate self-reacting B cells. And this really helps to minimize our risk of autoimmune diseases. So once again, if they do react with our body cells, we are going to go through apoptosis. And then we have our naive mature B cells now go to the spleen, and they're going to wait there until they come across a pathogen or another antigen presenting cell that has a pathogen's antigen to be presented to it to become activated. Let's take a look at our B cell receptors. These cells, remember, are going to be one of our antigen presenting cells. But what's interesting about it is that it can be activated without the help from T cells. So each of our naive mature B cells are going to have about 10,000 B cell receptors on its membrane. And they will be membrane bound monomeric forms of IgD or IgM. And within one B cell, each of these BCRs have identical epitope binding specificity in their antigen binding site areas. And just like T cells, we've used genetic rearrangement in order to create this. Now again, what's unique about these B cells is we don't need antigen presenting cells. They can recognize the epitopes associated with different molecular classes that they have, and they can interact with those epitopes or free antigens or with epitopes that are displayed on a surface of a pathogen. Now, the type of antigen we have is going to determine which process we're going to go through as far as activating the B cell. Our T-dependent antigen is typically going to be a protein antigen that requires our B cell to be an antigen presenting cell and place that epitope on its MHC2 to then present to our helper T cells. Our T-independent antigens will include polysaccharides, lipopolysaccharides, and other non-protein antigens that can activate the B cell without antigen processing and presentation to those T cells. So let's look a little bit more in detail of these processes. So when we have T cell independent activation of our B cells, remember this would include those antigens being polysaccharides or lipopolysaccharides, something that is non-protein. And so it's going to allow for cross-linking of multiple BCRs as their first signal of activation. The second signal comes with interactions with toll-like receptors, with PAMPs, or interactions with factors from our complement system. So once that B cell is then activated, it's going to go through clonal expansion and turn into plasma cells. Our plasma cells then are going to be able to create antibodies in large quantities and be able to secrete our pentameric IgM first, which will continue to rise, and eventually we'll get some antibody class switching and IgG will come in. And so these types of B cell activations are short-lived, and unfortunately no memory B cells are produced. So we will have to go through the same process if we were to be infected with the same pathogen again, and it would take the same amount of time. Whereas moving into our T cell dependent activation of our B cells, we go through this process when we have protein antigens. And the way this flows is we have to have that antigen on the bacterium and or any other pathogen, and we have to internalize the antigen and put it out on our MHC class II molecule and achieve link recognition, where we're going to have our T cell receptor of our helper T cell recognize this foreign antigen, and then the CD4 molecule is going to be able to attach to the outside of that MHC class II molecule on the B cell. At that point, our B cell is activated and our CD2 cells are going to produce cytokines that'll activate our B cell to go through clonal expansion and proliferation. And we will get activated clones of our B cells and have them turn into plasma cells, secreting lots of antibodies. And the other clones will create our memory B cells so that we create an immunological memory. That's why we always say this type of immune response is stronger and will allow for better recovery upon secondary exposure. We're also going to have class switching take place where we have IgM be secreted first, and then we will have IgG, IgA, and IgE with the same epitope specificity. Now let's review our primary and secondary responses. That T cell dependent activation of our B cells is going to really play into this secondary response here. Now notice when we have our primary response, we have this lag or latent period in which no concentration of antibodies are being detected. And this typically lasts about 10 days. And in the background, what's happening is our naive mature B cells bind to the antigen with their BCRs. We have antigen processing and presentation taking place. Our helper T cell and B cell are coming together and we're activating, and we get clonal expansion. And so as that is taking place here in the lag phase, then we will be able to start producing IgM. And this peaks at about 14 days after we're exposed to that antigen, and our helper T cells are then going to stimulate our antibody class switching. So we start to see a decrease in IgM, but an increase in IgG, which typically peaks around 3 weeks into our primary response. Now notice we, after a while are going to come back down to baseline. When we are exposed to that same antigen or that same pathogen, we get our secondary response. And here we're going to see that we more quickly and forcefully are going to be able to respond due to our memory cells that were created in our primary response. And the other thing to notice here is that within a few days, we are able to rapidly produce lots of IgG. And what we find is that this response is more effective and antibodies bind more aggressively and with higher affinity to the targeted epitopes. And we could see, especially with IgG, they do bind and stick around a bit longer in that secondary response to make sure that we are back to baseline as far as our health.